Hello everyone. So in this session, we'll be looking at biological databases and resources as part of the Introduction to Databases and Resources module. So before we begin with the lecture, let's have a look at some of the learning objectives for this session. So by the end of this lecture and practical session, we hope that you'll be able to understand how bioinformatics data is stored and organized, describe the different types of data found at the NCBI and EBI resources, and locate key bioinformatics databases and resources. In terms of practicality and practical outcomes for this particular session, we hope that at the end of the lecture and the practical session, you'll be able to understand the structure and layout of the NCBI and EBI data resources, understand the differences between databases, tools, and repositories, search for data from specific databases using accession numbers or gene names, and use some of the selected tools at the NCBI and EBI. So a big part of bioinformatics is essentially being able to analyze and extract and access data. In most instances, you'd either be using data that is stored in an online repository or generating data yourself and then trying to analyze the data. So in terms of data that's out there, there's a large variety of different types of data, and it's important to understand how this data is stored and where the data is stored. It's also important to understand what tools are available for you to be able to analyze your data in the case where you are generating your own data. And a big part of bioinformatics is understanding the concept of big data and big data management and access. So to start off, there's a large range of different online databases and resources in the bioinformatics arena. And in order to start off any project, or be able to start off any bioinformatics analysis, you first need to know what databases and resources exist, what tools are available to mine these resources, and then what tools are available to search across resources. And it'll become clear to you later on in the lecture that it's important to understand what systems there are or what tools are available for you to mine as many resources at the same time as possible, in order for you to save time when you're doing any sort of searching for data or any analysis online. So if you have a look at, or you try to find out what biological databases are out there, you'll realize that there is a large number of databases, resources, and tools available for doing different types of bioinformatics analysis. And it can be quite daunting when starting off trying to select the correct tool or the correct resource for you to extract data from, and also the correct tool for you to run your data analysis. So the first step is to have an idea or have a good indication of what databases and what resources are out there in order for you to try and select the correct database to use for your particular analysis. So one of the best places to sort of have an idea or to get an idea of what databases and resources are out there is two publications that are published by the Nucleic Acids Research Journal on a regular basis. So what the Nucleic Acids Research Journal does is at the beginning of every year, they have a special issue that is published, and this issue deals with particular bioinformatics databases that have been developed and are available online. An important aspect of the Nucleic Acids Research Journal is that they allow you to keep up to date with the latest resources that are available online in terms of databases as well as tools. So as I said before, in January they publish a database issue, and what they have in here is all the databases that are available online for bioinformatics storage of data. And what they do is they categorize all of them according to different types of data. So example, nucleic acid sequence and structure and regulation, protein sequence, structure, motifs and domains, metabolic and signaling pathways, etc. So if you're interested in a particular area of research and want to know what databases are out there containing information related to that area of research, this is probably your best place to look in order to find the most up-to-date databases that are available. In July of every year, the Nucleic Acids Research Journal also publishes what they call a web server issue. And the web server issue gives you a list of web tools that are available for doing different types of data analysis online. Again, this is a good starting point to go to if you are looking for an online tool or a standalone tool that will allow you to do a particular or to run a particular type of data analysis. So I would suggest that if you're starting up any research project that you have a look at these two issues and see what databases and what web servers are available for you to do your particular analysis. The good thing about the Nucleic Acids Research Journal publications on databases and web servers as well is that it allows you to make sure that you're using the most up-to-date resource. So what they do is for each of the databases and resources, they publish a paper related to whether that database is still being updated and maintained, as well as the web servers and whether they are still being updated and maintained. So an important concept to keep in mind as well when you're doing any sort of bioinformatics analysis or retrieving data from databases online is to ensure that you are always retrieving information from an up-to-date resource or using a tool that is regularly updated and maintained. So let's have a look first at databases. So in bioinformatics, you'll find that most data is stored in a database. And there are some different aspects or different types of databases that are available out there. So the first is that some of the databases can either be public or private. Luckily, in the bioinformatics arena, majority of databases are public, and therefore the access and as well as submission of data to these databases is usually open to the public, and there's no need for you to pay anything or to have a subscription to be able to access this data. We also do have some private databases online, and in most instances, these databases are housed and maintained and generated by private companies and used for development of products. In terms of databases being public or private, this is in terms of access and submission. So you might find that some databases allow you to publicly submit data to these databases, but might have private access and vice versa. In terms of different types of databases, you'll find that there's different databases for storing protein information, protein data, nucleotide data, structure data, literature data, annotation data, etc. And we'll have a look at some of this in more detail in the rest of the lecture. A database can then also be generalized or specialized. So an example of a generalized database would be, for example, a database that just stores nucleotide sequences. So an example of this is a database at GenBank, which we'll speak about later, but it's essentially a database where you're able to submit any nucleotide sequences that are generated for any organism. We then also have specialized databases. So these might be specialized databases in terms of a particular pathogen, a particular organism. It may be specialized in terms of the type of sequence data that's added to it, etc. And we'll speak about this a bit more later on in the lecture as well. We then also have the concept of curated versus non-curated databases. And this is an important concept because it is important to understand what is the quality of the data that you're accessing and how has this data been checked or validated. So in terms of curated databases, these are databases where the data has been submitted, maybe publicly by anyone. However, the data then goes through a process of being checked. And by checked, it means it is physically checked by a human individual who goes through all the relevant information that should have been submitted with the data to make sure that everything is correct. Now, in terms of the data deluge that is part of bioinformatics, and by data deluge, I just mean the large amount of data that you're able to generate quite quickly and easily these days, there are also many non-curated databases. So these are databases where the public or scientific individuals are able to submit sequence data in most instances to a database or annotation information to a database. But the data is then not curated. So that means that it is not manually checked to ensure accuracy of the data. And you'll find that there are large amounts of bioinformatics databases that are non-curated. So it's important to consider this when you are accessing data from the database, whether it is a curated or non-curated source. And then we have databases that are either sequence or genome-centered. So sequence databases will be databases where you just have sequence data that is submitted and you are able to only access information from the database if you know a particular identifier for the sequence or a gene name for the sequence. But then we also have genome-centered databases or genome-centered resources, where, for instance, you'll be able to access all the information for a particular genomic region in a particular organism if you are interested only in a particular region of the genome. So for example, say you're interested in identifying all the genes on chromosome 1 in human, a genome-centered resource or genome-centered database will allow you to query that database saying, I would like only the genes on chromosome 1 in human. And we'll have a bit more of a look at that in the lecture as well. So in terms of different types of databases, the first and most simplest type of database is the primary databases. And the primary databases essentially store the most raw version or the most raw, essentially the raw version of the raw data that's available. And in terms of bioinformatics, these primary databases mostly store sequence information. So this can be nucleotide sequence information or protein sequence information. In terms of protein sequence information, we have primary databases that store raw sequence data for proteins, as well as structural information for proteins where the structure has been solved. So the raw sequence databases are essentially where the primary sequence databases is where the raw sequences are stored, and then on top of these, we'll see that there is additional databases where we have annotations built in. So in terms of primary databases for sequence information, so essentially what happened was at the beginning of sort of the sequencing era, where sequence data was being generated across different organisms, there was a need to form a formalized way in which sequence data was stored, was stored and accessed. So this led to the formation of the International Nucleotide Sequence Database Collaboration, INSDC. And this was a collaboration between 3 separate institutes. And essentially it was a genomic sequence data, it was a way in which genomic sequence data could be stored in 3 public databases. And these 3 public databases would then communicate with each other and update each other on a regular basis. So the first is DDBJ in Japan, the second is NCBI in the United States, and the third is EMBL or EBI in the UK. And each of these, so what happens when you generate sequence data, you're able to submit that sequence data to any one of these individual databases. And there's regular updating between these individual databases on a regular basis. So each have their own accession numbers and tools. However, they are linked between the 3 databases. So on top of the primary databases, we have secondary databases. And essentially, these are more in-depth databases in which information is built upon the primary sequence data. So an example of this is where we have annotations for a particular sequence. And there's several different types of secondary databases whereupon we have annotation information that is then linked to the primary sequence data. So 2 of the most popular bioinformatics resources where data is stored the first is the National Center for Biotechnology Information, or NCBI, in the United States. And the second is the European Bioinformatics Institute, or EMBL-EBI, in the UK. And what we'll be doing is we'll be having a look at in more detail at these 2 resources in the next session.